In this third section, we are going to talk about first about muscle proteins. And to talk about muscle proteins, we are going to talk about three types. The contractile, that are the most famous one that you have here, actin, myosin and actin. Then we are going to talk about the regulatory, that are the one that uh, regulate the muscle contraction by affecting actin and myosin in different ways. And finally, the structural, that are the one that make that the proper alignment of the actin and myosin take place, and the muscle have some elasticity. Later, we are going to talk about sarcomeres. And sarcomeres, and here I'm going to show you this picture all the time, is just the unit of contraction. So all these proteins that we talked in the beginning will be located here. And why they have these colors, this structure, is because the actin and the myosin and all these other proteins are located there in a very organized way. So we are going to discuss why you have one region that look white, while ones that are look darker. Yeah, and why you have lines like this. And we will describe all these bands and this and lines, okay? Finally, at the end, we will talk about the sliding filament mechanism. And in your textbook, they have one wonderful video that describe all of that. I will post a link so you can watch it. Here, I'm going to give you a very quick explanation of only 4 steps. Okay, I hope that you're excited to finish this section. So let's start right now. Now, let's start talking about the first point, the muscle proteins. So when we talk about muscle proteins, you should know that there are 2 that are very important. One that is actin, that is found here. That is the thin filament. That is the yellow one, and we're going to show this schematic multiple times. And the other one that is the myosin, and the myosin is the thick filament. Okay, they have different structures. So here, for example, the myosin had a head, had actin binding sites. If you see attached to the actin, you also have other proteins that are the regulatory proteins, and we will talk about that in following slides. So from a very general perspective, for sure you need to remember that there is a group of proteins that are in charge of the muscle contraction that are myosin, are the thick filaments that you can see here in red, that are in charge of the power stroke. So that is important that you remember it. And the actin are the thin filaments that you see here in yellow. And those filaments are in charge of sliding, yeah? That we will see that at the end of this section while we talk about with power stroke and sliding. So let's start with the contractile proteins, and the contractile proteins are the ones that are in charge of generating force during muscle contraction. And there are 2 types, myosin, which is the first one, and actin, and I will keep repeating this. Myosin is the one that is in the thick filaments. So now let's go and see in more detail this schematic of your textbook. So when you see this schematic, you see that there is not one molecule of myosin, you have multiple molecules. And if we take only one, we can zoom in into only one. And there are 2 sections, the tail, one tail, and you have 2 heads. Here you see the 2 myosin heads, and the most important part in the myosin heads is that there are binding sites, one for actin and one for ATP. So if the myosin is attached to the actin, it's not going to be attached to the ATP and vice versa. So this is why we need we are constantly spending ATP during muscle contraction to attach and detach to the actin and produce the sliding that we will see in a second. Okay, so remember, the molecule of myosin is formed of 2 sections. One that is the tail that you see here in green, and the other one is the head, and you have 2 heads. And inside of the heads, there are the 2 main components that are the binding sites. one for actin and the other one for ATP, and they are very important for the muscle contraction. In this slide, I'm going to talk about the second contractile protein, that is actin. Here I show you this schematic of your textbook that I will show you again multiple times. In yellow, you can see the molecule of actin. Here it is. So all these circles are representing the molecule of actin. And remember, the contractile protein, that is the main component of the thin filament, is actin. There could be more proteins, but this is the main one. And if you see the other ones that we will talk in the following slides are troponin and tropomyosin that are in charge of regulating myosin, because myosin has, if you see here, here is actin, and here is the main component that we need to pay attention, is the binding site, the myosin binding site. So the myosin is going to bind here, and if you see, the tropomyosin is going to try to cover it. Yeah, so if there is calcium, the tropomyosin is going to move, and it will give a space to actin to bind, and that is very important for regulating muscle activity. Okay, so I told you that each molecule of actin has a binding site, so each one of these has like this tiny hole that is, in this case, covered by tropomyosin. And I told you that this binding site is where the head of myosin of the thick filament binds during muscle contraction. Now we're going to go into the second group of proteins that are the regulatory proteins that are in charge of helping the muscle to switch from on and off, so from a relaxed to a contracted process. And here you can see a schematic again with the two key proteins, that one is troponin and the other one is tropomyosin, and both are regulating actin. How? So if you see, tropomyosin is look like a rope, and is covering what? The actin binding site of myosin. Yeah, and what is troponin doing? Troponin is making sure that tropomyosin is in the right place. Okay, so these regulatory proteins are a component of the thin filament, and I told you about tropomyosin, that is very important, that binds to troponin, that is the blue one in this schematic. Troponin aligned to the binding site, that is very important, the binding site to the myosin. Now I'm going to show you a video that shows the interaction of troponin, tropomyosin, and actin, especially the actin binding site, and it shows how they regulate muscle contraction.
At rest, the active sites of actin are covered by a rope-like molecule known as tropomyosin, which helps to stabilize the actin and also blocks the globular heads from binding to these active sites at rest. Another molecule known as troponin holds this tropomyosin in place. When calcium is released from the sarcoplasmic reticulum, which covers the sarcomere, the calcium can bind to the troponin and move tropomyosin out of the way, which then opens the active sites of actin so that myosin globular heads can form cross bridges and initiate a contraction.
So I think that video summarizes everything. So remember, we have two molecules that are very important to regulate the muscle contraction. Tropomyosin, that we saw in the previous slide, that it looked like a run, and here the one that looked like a pearl, the circumference in blue. And as you watch in the video, when the muscle fiber is relaxed, so the troponin is properly attached into the tropomyosin, so the rope, the blue rope, is covering all this section. Then what happened is that when to turn on the muscle contraction, we need to remove the tropomyosin and the troponin. So how we do it? By releasing calcium, and now the troponin is not bind anymore to the tropomyosin, and here the tropomyosin. Let me see if I can draw something here. The tropomyosin is not here anymore. Yeah, it moves away, and as it moves away, now myosin can attach to the binding site. So remember, the tropomyosin cover the myosin binding site, preventing the myosin to bind to actin. And to remove it, we just need to have calcium to disassemble all these proteins that are blocking the binding site. Okay, here you have two type of schematics. In the right here, you have one complex that look very complicated. Don't worry about it. We at the end of the class and during the test, you will see that it will become much easier. And here, just an animation of what is happening just with the sliding of the filaments, of the thick and thin filaments. Why? Because I want to explain the last type of protein. So we already talked about the contractile protein that you can see it here, the thick and the thin filament of actin and myosin. We saw the regulatory ones. And now we're going to talk about the structural. And one thing that the structural proteins are doing is, if you see, the question is why the sarcomere doesn't go beyond this point, yeah, or doesn't go even closer, and why the sarcomeres align and they don't bend a little bit in this direction or in the other direction. Why? Because you have structural proteins that prevent that this sliding goes in the wrong way, yeah. So let me show you. So there are three key functions of this structural protein. So let's forget about the sliding. Let's remove it. And titin and dystrophin, these two proteins are in charge of help the sarcomere to be more stable. And now let's focus on the complicated diagram. So here you can see the titin that look like a coil. Let me show it to you in red. Here you see one example. Here you see more examples. So it looks like that this protein is going to be important for the flexibility and elasticity of the sarcomeres. So it's going to prevent that it goes too far or don't go or that it goes in the right way. So that it has the proper alignment. Also, we have another one, the dystrophin, that if you see it here in purple, you have one here and one here. One of the titin was connected with the actin and the filament, yeah. And in particular, look, it goes all the way up to here. The dystrophin, it is in contact with the sarcolemma, yeah. So if you see here, all these things are membrane proteins. The sarcolemma is the membrane protein, so it will be in contact with the extracellular matrix. So you can say like how much actin it can get, all these important things that it can is important for the muscle contraction that is communicating with the external environment of the cell. So just to summarize, I told you that I was going to give you three main functions. The first function that we talk is that the proteins, these structural proteins, allow to keep both thick and thin filament of myofibrils in proper alignment. So I told you the sliding cannot go in other direction. It has to go in the proper alignment. The second one that I give you is that they can work to give more elasticity and flexibility. In particular, here the titin is has a major role on that. And finally, the dystrophin has a major role in linking the microfibrils to the sarcolemma and the extracellular matrix. Okay, so those are the three function of the structural proteins. Now we're going to move into explain a little bit in more detail the functions of the titin and this function of the dystrophin. So we're going to talk about titin and how titin stabilizes the sarcomere. So if you remember, titin is these proteins, and if you see the structure, they show it like this, like a spring that is going back and forth. And this is one of the key functions, one of the key ways to give more stability and flexibility to the sarcomere because it can stretch and then spring back and harm. And let's talk a little bit about this complicated schematic that we have in the right. You don't have to remember all the proteins, but here later we will talk that there is an S, a zeta line, yeah, here, and this is the protein. You don't need to remember this one. I just wanted to show you that there are many proteins. We are going to focus on some of them, but if you end up doing a studying physiology of sports or something like that, or there is, I'm just summarizing the information. There are more complicated things. So it start here. You see it goes all, give all these terms and then continue and attach here to the M line. And in the M line, you of course you have another type of proteins, yeah, but the titan is attaching to both of those, yeah, so to the Z line and to the M line, and this allows them to have more control over the sarcomere, and in this way produce give stability, elasticity and extensibility to the myofibers. Okay, so remember this is the protein that structural protein that we are studying, and this structural protein titan connect to the S, to the Z disc, to the M line of the sarcomere. It also, this structure allowed to stabilize the thick filament position. How it does it? Because it can stretch and then spring back and harm, allowing to this sarcomere to be to have elasticity and flexibility at the same time. Okay, so the last protein that we're going to talk is dystrophin, and I'm sure that you remember that it's located here. It is the purple one, and is attaching the actin, the thin filaments, to the sarcolemma. You can see it here, and if you remember, sarcolemma is the cell membrane. So here the sarcomere is in the cytoplasm, and outside you will have the ECM, the extracellular matrix. In other words, the dystrophin stabilizes the sarcomere by making a connection, yeah, from the actin to the exterior, from the interior to the exterior of the muscle cell, and so it attach the thin filament and allowed to communicate it to the connective tissue here in the SEM and later to the tendons that they are going to be attached to the bones. Okay, so it allowed to have all these connections. So this dystrophin is the last structural protein that we are studying, and this one, instead of being stabilized in the thick and the thin filament, this one is attaching to the cell membrane of the muscle cell or sarcolemma, and this one in turn is attached to the connective tissue matrix, which help to transmit and generate the tension transmitted to the tendons, and in that way pull the bones. Okay, so we are done with all the proteins. I'm just going to add an extra slide for you. Here you can see all the different proteins that they were found in your textbook. If you want to study, I didn't talk about this one, or this one to not make it more complicated. And there will be practice tests, so you know what type of question and how much in detail you need to study all these things for the test.